Good morning, everybody. My name is Chloe. Oh, hi, Caitlin. And I'm a sophomore this year, and today I will be presenting my summer research for 2023. My mentors were Dr. Hayes and Dr. Young from the Bishop Museum. First, environmental DNA, or eDNA, is organismal DNA that can be found in the environment. So it can be found from damaged tissue, metabolic waste, skin and scales, and free DNA. So essentially, wherever a living organism goes, it leaves some trace of itself behind. And in the case of seawater, these are often ways that it gets into the water. And my goal is to essentially take this DNA and analyze it. Protocols using eDNA may allow for rapid, cost-effective, and standardized collection of data about species distribution and relative abundance. As you can see by this flowchart, it has many different steps, but I'll go into that later in my presentation. So to get into why it's important that I do this, so the biodiversity of islands around the world is becoming increasingly threatened due in large part to habitat loss, overexploitation, invasive species, and climate change. So this specifically is really important to us because we live on an island and it has a really interesting diversity of species. Therefore, it's really important to protect it because we have a lot of native species that are being outcompeted by invasive species. So I personally also took a liking to intertidal zones and algae. So I've always wanted to do a presentation or like a project on it. So this is my chance to, I guess. I also learned a lot about overexploitation, invasive species, and climate change in my science Olympiad testing. So yeah. To go more into depth about why invasive seaweeds is bad is invasive seaweed smothers coral and degrades the environment needed by native fish, invertebrates, and lemur. So essentially what these invasive algae will often do in an ecosystem is they will overtake or literally grow on top of the native algae, and they will like shield the light from the native algae. Therefore, the native algae die off because they don't have light to perform photosynthesis. And the invasive seaweed pretty much takes over the environment. And this affects the native fish and invertebrates because these organisms relied on the native algae rather than this new invasive algae. So they may have problems regarding their food source, which could lead to the detriment of the entire native ecosystem. For my research question, what is the distribution of native and invasive algae in Haleiwa using eDNA? For my hypothesis and my purpose, my hypothesis is the distribution of algae within Haleiwa Beach Park will contain mainly native algae, as it is kind of commonly known that on the North Shore, it generally should be composed of only native algae. And of course, my purpose is to fight invasive species to protect native ecosystems. So to go into my methods, sorry. First I took seawater from Haleiwa Beach Park. So I went out with Dr. Young and Dr. Hayes on a BioBlitz type situation with some UH interns, and we went over to do some sampling for the Bishop Museum, and there I also got my seawater to do my project. So for each sample, I gathered 500 milliliters of seawater and pushed them through an apparatus which would pump the seawater through a 0.45 micron filter. And on that filter would essentially collect all of the DNA that I wanted to save. And at the bottom of the apparatus would be the filtered seawater, which I didn't really need. The most important thing to me was the filter because that contained the DNA. We stored this filter in a solution called Longmire solution, which essentially makes it so that the DNA is less likely to degrade, or because because it can be a problem in eDNA if you don't have something to hold the DNA in, or hold or suspend the DNA in, it could degrade really quickly. Therefore, you wouldn't have anything to sequence or match to libraries, which I'll get into later. And my next step was extracting DNA. So we used a plant OPS kit to extract this DNA, what essentially we did was we took away all of the non-DNA particles and we focused on having, I guess, a supply of DNA that we wanted to use to compare against different types of algae. So next, it was about cleaning up this DNA and amplifying it, specifically the barcode region, specifically the barcode region. So the whole way eDNA works is you look at this series of ACGTA and you identify that to a database because databases have it sequenced and they have the species that it belongs to. So first we use PCR to amplify this barcode region and basically create a bunch of copies of it. So we had a little bit of contamination when it came to PCR. So we had to try many different methods to try and get rid of that contamination. But eventually we got it, so I was able to get results. Next, we sequenced the DNA and matched it to reference libraries. So I worked with a group of people who were also working with algae and eDNA, Ellie and Maddox. So together we created this library of DNA sequences that we could use to match what we found in our DNA versus the DNA that was in those libraries, so we could see the different types of native and or invasive algae that were in our samples. And finally, we were able to basically take all of our DNA and match it against those reference libraries, so we were able to get results. So despite problems with contamination, we were able to amplify and sequence one filter sample. So you can see it's labeled PCR number 1. We can see we have some contamination of our PCR negatives, which is not too good. So we decided to do it again, and you can see in PCR number 2, we had worse contamination, which is not great. But eventually in PCR number 4, we got it down to, or our PCR negative was in ladder 1. And as you can see, there's not, it isn't distinctly like dark like the rest of like our ladder or our samples 2 and 3. So this was a good sign, and we used this to essentially know that our DNA was somewhat not contaminated, and we moved into the stage of basically taking the sequences and matching them against the libraries. So we amplified both algae and fungi DNA. Our total number of classified reads was 2,416. So this would just be the amount of recognizable sequences that we had in our entire project. So as you can see, we had a really big distribution of different things, but importantly, most of these like large pie pieces were fungi because of most likely contamination. But we got algae, so that's the part we focus on. Most algae reads were Padina, and our total number of algae reads was 295 out of those 2,000. So if you take away all of the fungi readings and you just put the algae readings in a pie chart, this is what it looks like. Some algae that are notable here are the Padina australis and Melanomancia glomerata, which are both native species that we like to see in the environment. So it's really good that we saw that. We also saw Gracilaria salicornia or gorilla ogo, which is invasive, which is not as promising, but 96% of our reads were native, which is really important because, as I said earlier, the North Shore should be mainly composed of native algae. So this is really good news. And I have some more pictures of native algae here. So for my next steps, probably more trials and research around the North Shore in order to devise a plan to protect the native species of algae, because we don't want another ecosystem being taken over by these invasive algae. For example, Kaneohe Bay specifically was overtaken by an invasive algae, and now there isn't really any traces of native algae there. So it's important that we protect the ecosystems that we have. So for my reflection, what did I learn? I learned a lot of problem-solving skills and more about the native intertidal ecosystems of Hawaii, which was one of my goals, so I was really happy about that. And I learned how to work with others and apply my basics in science to learn popular research methods. So what I learned in biology, for instance, last year, I could use to understand what I was doing over the summer. And my advice to other students, don't be afraid to ask questions or get things wrong, and learn to build good connections with others. So for some acknowledgements, I have Dr. Chan, Miss Kobayashi, and Mr. Hill, because you guys helped me out a lot when I didn't understand what I was doing. And you guys gave me a lot of good advice about how I should structure my project, and if I needed to make any changes, what would be the best way to implement that. And I also have Dr. Young and Dr. Hayes from the Bishop Museum, as we use Longmire solution from them, and they also took me out to Haleiwa Beach Park. Thank you.